Hello, ladies and gents. I'm sorry, couldn't resist the opportunity there. Hope you're doing well today. What we're going to do now is use that big old piece of paper you got and make a giant diagram that sort of walks through all the ideas related to Chapter 9 over cellular respiration. So, first thing we're going to do, we're going to use a black marker, and I'm going to fill this dry erase board. When I do that, that's in essence the same as filling your big old piece of paper. So, be brave, be bold, fill up the entire space. So, what we'll do is use almost this whole space. And again, we're going to make just a giant cell, world's biggest cell. All right. And when we have that cell, we're just going to label it, well, cell. Inside the cell, try to see if you guys can see that. I think it comes through okay. Inside the cell, we're going to label it cytosol to indicate that fluid inside the cell. Then we need to make the mitochondrion. Now, this is going to be pretty massive, so the mitochondrion is going to take up a majority of the space on the board. Again, it can make you uncomfortable, but I would say more than half, maybe 2/3 of the page should be your big old mitochondria. So I'm going to be using brown for this, and I'm going to again have this take up at least half the space. So there's the outer membrane, and then we need to draw that second inner membrane. Okay. So now let's label the parts of the mitochondria. So we'll just label the whole thing, and we'll label that mitochondria. Let's see. Then we have the inner membrane. The inner membrane is called the cristae, C-R-I-S-T-A-E. So if you want to have sort of a unique spelling for a child's name, Christy with an A-E might be a way to go. Now, inside the mitochondrion, that fluid is called the matrix. And then the space between the cristae, the inner membrane and the outer membrane, is the imaginatively named intermembrane space. I think it's a wasted naming opportunity, but it is what it is. This could be the Ray Skillman intermembrane space. All right. So there we have the basic setup of the mitochondria. Now, we know that cellular respiration happens over the course of a few different steps. Okay, so what I'd like to do is begin with our original energy molecule, and that is glucose. Now, the way I'm going to represent glucose, I'm going to use the black marker to make some dots to represent the carbon molecule, or the carbon atoms. 2, 3, 4, 5, 6. So I've got 6 carbon glucose. And then I'm going to draw red between them to sort of symbolize the energy in the bonds. They always draw like zigzaggy lines to represent energy and stuff, right? So here we have our 6 carbon glucose, and let's see if dimming the lights helps at all with the glare. And I don't think that helps too much. Dimming that blank, that might help with the glare just a little bit. All right, so we have the 6 carbon glucose here. Why don't we label this glucose? Now, glucose will be transported inside the cell. And then let's redraw the glucose. Okay. And then glucose will go through the metabolic process of glycolysis. So we'll draw an arrow here. This is glycolysis. And at the end of glycolysis, we break and rearrange glucose and end up producing 2 molecules of 3-carbon pyruvate. So we'll show the bonds between the pyruvates. We'll label that pyruvate. All right. Also from this, we're going to get a little bit of energy production. So we'll get a couple of ATP and we'll net a couple NADH. There's some energy. So when glycolysis occurs, you'll net the production of a couple of ATP. You'll also strip away some protons and electrons, and you'll wind up sending those to NADH. All right. Now, most of the energy in this instance is still locked up in pyruvate. So what we need to do is continue to break that down. So we'll just take one of the pyruvates for the sake of simplicity, and we'll move that over to the matrix of the mitochondria. So I'll take that 3-carbon pyruvate, and then if you'll recall, it undergoes that link reaction. Now, the link reaction is going to split off the carbon dioxide. It splits off a carbon, and that carbon dioxide will end up getting released from the cell. So some of the carbs that you consumed, you'll break them down with the link reaction, and that CO2 that's created, you'll blow that out. All right. What you're left with is a 2-carbon molecule. And then this 2-carbon molecule can get fed into the citric acid cycle. All right. So we can say here is the Krebs or citric acid cycle. So here's our Krebs or citric acid cycle. And what you do is you feed this 2-carbon molecule into the process. And this 2-carbon molecule will attach to an already existing molecule, like oxaloacetate. You don't need to know any of that. Just know that the 2-carbon molecule gets attached to an already existing molecule. Now, what happens with that is all sorts of rearrangements. There's a whole bunch of reactions that go on here. You don't need to memorize any of the enzymes, any of the intermediate products. All you need to know is sort of the general idea of what happens. What happens is you split off some of the carbon dioxide. So what ends up happening is you completely break down the 2 carbons that came from originally pyruvate, which came originally from glucose, which came from your meal. So you consume your food, it goes through reactions in the Krebs cycle or citric acid cycle. Those carbons get split off, and then you exhale it. So sometimes what's said is what you eat, you breathe out. Some of the byproducts of what we eat, we actually exhale and put back into the atmosphere. Also in this process, you strip away some protons and electrons. So those electron carriers like NAD+, they'll get used to generate NADH. And FAD, another electron carrier, becomes FADH2. Also in this process, you end up generating a couple of ATP. And you're only netting a couple ATP, not enough to live on. Well, at this point, we've completely broken down your food. You've taken your 6-carbon glucose, you split it into 2 molecules of pyruvate. The pyruvates will get fed into the matrix of the mitochondrion. They'll undergo link reactions. You create these 2-carbon molecules that attach to an existing molecule in the Krebs cycle. Enzymes cause all these conversions. You strip away those carbons, they'll get exhaled, and in the process, you net some ATP. Now, all along these pathways, again, you're stripping away protons and electrons. So at this point, you've created a number of NADH and FADH2 molecules. So you've got energy that's bound up in these molecules. So now what we're going to do is move them on to sort of the last set of events. That's the electron transport chain and oxidative phosphorylation. So let's see. Let's add some stuff to our Christie, this inner membrane. So I'm going to add some proteins in here. Some shuttles. Okay, so here, ooh, I hope it's not blocked too badly. Oh yeah, sorry, let me see where I'm at. This faucet completely blocks ATP synthase. So let me move it. Let me draw another one here. Right about here. Oh, that's coming through, I think. Okay. Hopefully you can see that. And we're going to make it work, okay? So anyway, here are the proteins of the electron transport chain. And then here is our hero, ATP synthase. All right, so now what's going to happen is NADH, FADH2, they're going to carry their electrons to the electron transport chain. Now, a couple of important things happen. First, protons are removed from NADH and FADH2 and then put in this intermembrane space. So now we're creating a gradient. We're concentrating protons in this space. Let me add some down here as well. Goodness, there's a lot of protons. Okay, so then what will happen is these protons create a gradient. All right, now With ATP synthase, the protons are able to flow down their chemiosmotic gradient. They go from where they're higher ion concentration to where they're a lower ion concentration. When that happens, it makes ATP synthase spin. So I got an arrow here to try to indicate that it rotates. When that occurs, what it does is it brings together ADP. Can you see that? Okay, it brings together ADP and an inorganic phosphate. That's just a little phosphate by itself. And then that ends up creating a bunch of ATP. And not just a little bit of ATP, lots of ATP, a whole lot of ATP. Okay? So this is the process of oxidative phosphorylation. When you have these protons flow down their electrochemical gradient, it spins ATP synthase, you bring together ADP and phosphate, and then you make a whole bunch of ATP. Now, I've still got something going on here. Not only do NADH and FADH2 carry protons, they also carry electrons that get handed off to the electron transport chain. Now, these electrons get passed down the electron transport chain. And what you wind up with are increasingly electronegative protein complexes that draw their electrons or draw the electrons toward themselves. Now, at the end of this, you got these low energy electrons. And low energy electrons, they're just like any of us. They just want to be accepted. Okay? So what's going to accept those low energy electrons? It's going to take a pretty electronegative atom, and that is oxygen. So the oxygen we breathe in, I'm going to try and have it swoop in here from this direction because we're sort of tight on space. But the oxygen that we breathe goes in, accepts these low energy electrons, and combines with the protons that pass through, and it forms H2O. Okay. So the oxygen we breathe in combines with electrons from the electron transport chain and protons that flow in, and it forms water. So earlier we said what you eat, you breathe out as CO2. It's a little crude, but sometimes what people say is what we breathe in, this oxygen, we pee out, forms water. Okay. So now let's try and tie all this stuff back together again. We start off with glucose, a high energy or a carbohydrate molecule. Inside the cytosol of the cell, it goes through the glycolysis reaction. It gets converted into 2 molecules of pyruvate. The pyruvate then gets passed on to the mitochondria. In the mitochondria, it undergoes a link reaction and splits off carbon dioxide, and then it forms a 2-carbon molecule. The 2-carbon molecule then joins the Krebs cycle. It gets attached to an existing molecule. And in this process, it'll get rearranged. You get some carbon dioxide split off. Again, you're going to exhale your breakfast. And you'll strip away some protons and electrons and form NADH and FADH2. Those molecules, the NADH and FADH2 created during glycolysis and Krebs, they go to the electron transport chain. Then at the electron transport chain, the protons from your food create this gradient in the intramembrane space. The protons flow back through ATP synthase, making this enzyme spin, and it combines ADP and phosphate to form ATP. You get a lot of ATP, like say 32 ATP per molecule of glucose. Let's see, the electrons get passed down the electron transport chain and then eventually accepted by the oxygen we breathe in. The oxygen accepts the electrons, combines with protons, and then it forms water. So in this process, we're able to break down the bonds of food, strip away protons and electrons, and ultimately end up spinning an enzyme that brings together ADP and phosphate in order to make ATP. Now, what if a person doesn't have oxygen? If a person is lacking oxygen, then you don't accept the electrons into the electron transport chain. And what you end up doing is shutting down electron transport. That backs up the Krebs cycle, and then we're just left with glycolysis. Glycolysis doesn't require oxygen, so this can run without the presence of oxygen, so long as we can regenerate the NAD plus that will be the electron carrier. So that's when you get into like lactic acid fermentation, stuff like that, that you can reference back from the lecture. All right, so these are the big ideas. If you have questions, let me know. Otherwise, I wish you the best of luck, and I'll see you soon. Here's the awkward part that I don't know how to edit out easily.